All right, here we go with section 3.4, looking at the different internal structures of a bacteria cell. And within chapter 3.4 of your textbook, there are individual subsections, basically outlining the different major features of the internal structure of the bacteria cell. For the lecture notes, though, we're just going to hit on a couple of them. The first is going to be the cytoplasm. All right, the cytoplasm is actually 70 to 80% water. So you think about it similarly in terms of our Earth. Our Earth, the Earth, is a planet, and it has different components of the planet, but 70% or so of the planet is water. All right, cytoplasm, 70 to 80% is water, and then the remaining are going to be different macromolecules that are coming together to produce or to have a specific function. So there's going to be the proteins, there's going to be salts that are going to be dissolved within the water, and then different carbohydrates. And the cytoplasm is going to be the site for nearly all chemical reactions, except for ATP generation, because if you remember from the previous section, that's going to occur within the plasma membrane. And within the cytoplasm, there's going to be a major area that has the DNA, and that area specifically within the cytoplasm is called the nucleoid. Remember, because these are prokaryotic cells we're talking about, by definition, a prokaryotic cell does not have a nucleus like eukaryotic cells do. All right, then just moving directly into the bacterial DNA, all right, it's genetic material. It has to have genetic material in order to pass on anything onto the next generation. All right, so the DNA of most bacteria exists in the form of a single circular chromosome. Human chromosomes are long pieces of DNA, and so you have chromosome 1, chromosome 2, all the way up through 21, and then the chromosomes X and Y. And if you see a picture of them, they kind of look like an X, all right, because you have two straight, relatively straight homologs. All right, with bacteria DNA, it's going to be circular. So the DNA is going to be aggregated or basically condensed into one area of the cell, and that region is called the nucleoid. Now, another piece of information that I mentioned when we talked about conjugation or the pili as an external feature of gram-negative bacteria. All right, some bacteria will have plasmids, and these are going to be very small circular pieces of DNA that contain non-essential genes. Right? Non-essential means it's not critical for survival. So a lot of times the genes that are present on a plasmid are going to be pathogenic genes. Let's say for it to express some sort of antibiotic resistance character traits, or to actually be able to produce a pili. All right, being able to produce a pili to pass DNA onto its neighboring cell isn't necessary for the survival of the bacteria cell. It's still going to go through replication and be able to reproduce. So that's what it's talking about with non-essential. It's generally pathogenicity factors, and an organism can still survive even if it's not pathogenic. All right, then another major feature is endospores. All right, a lot of times bacterial endospores get confused with fungal spores, but the bacterial endospores, and you have to have that endo in there. These guys are going to be dormant cells, dormant bodies that can survive for years, or I don't know if thousands of years is correct, but hundreds of years, centuries. and they are going to be resistant to heat. And the reason for that is that because the outer shell of that endospore is going to be filled with calcium and dipicolonic acid, and that makes the endospore very strong, and again resistant to the heat, resistant to very dry conditions. Whereas if a bacteria cell was out in the environment and it was a drought, right, 80 to 70% of the cytoplasm is water, and that water is going to eventually leave the cell due to evaporation and exposure to the elements, and that bacterial cell will die. Well, if the bacteria cell can produce an endospore, then it's actually going to be safe in those very harsh conditions. All right, there's also part of that outer layer of the endospore, the cortex. This is the spore coat, and it will protect against radiation and chemicals. And so if you have endospores that are floating around in the air and they get into a hospital setting, sometimes radiation and chemicals are used to try to decontaminate or even sterilize equipment. But if you have endospores present, a lot of times those features, those processes aren't going to work. So, metabolically active vegetative cells, so a live bacteria cell, can undergo the process of sporulation. This is the process of creating that endospore. All right, sporulation is not a reproductive function. Right? It's not doing this so that it can reproduce itself and continue on life. Right? But rather, the vegetative cell, the metabolically active cell, realizes, senses that the environmental conditions are getting poor, the nutrients are going away, the moisture is going away, it's getting too hot. It can go through that sporulation and make itself safe. Right? You can kind of think about it as a forest fire, and a firefighter being in the forest fire trying to get rid of that fire, help fight the fire, and get overtaken by the fire. And so what is that person going to do? He or she's going to try to survive. They're going to grab their fire blanket out of their backpack and cover themselves completely with the fire blanket, get down to the ground, get as flat as possible, hope that fire goes over quickly so that they can be safe. Right? That fire blanket for the firefighter is very similar to the endospore for the bacteria cell. Now, when these endospores, all right, see here it says spores, and that could be misleading. It really should say endospore. When the endospore of, say, Clostridium are embedded into a wound, and really a wound with dead tissue, the endospores can then germinate, move back to a metabolically active vegetative cell, grow and release toxins. So Clostridium tetani, Clostridium botulinum. These are organisms that can produce endospores, and then you step on a dirty, let's just go with the most obvious thing, a dirty nail, a rusty nail. And what happens? All right, a person could potentially get tetanus from that. Well, it actually has nothing to do with the rusty nail, but rather the rusty nail has a lot of dirt on it, and there's a lot of Clostridium in dirt. There are endospores in the dirt. And so what does that rusty nail do to the tissue of your foot? Well, you step on it, it goes into the tissue. Because it's rusty and all nasty and hard and very rough, it makes a very nasty tissue wound, right? It's not like getting cut with a razor blade, and it's a very clean cut. And you can open it up, clean it, close it, wrap it up tight, and it heal very easily. With a rusty nail, you kind of make, hamburger meat, lunch meat out of your tissue. And it's very difficult then to try to open that wound up, get cleaning solution down in there, but rather you've got the tissue that comes back together, but there's dirt underneath that tissue. You just can't clean that wound well. And so then if you've got dirt in there that has the endospore in there, you can have a very nasty wound a few days down the road. All right, so here's the process of sporulation. All right, here you have the vegetative cell. All right, looks pretty normal. All right, but now this cell begins to recognize that the environment is becoming hostile. The nutrients are being depleted, too hot, not enough moisture, whatever the case may be. All right, so it senses that, and if it's a cell that can go through sporulation, it begins that process. It's going to replicate the DNA. The DNA is going to become separated to different poles of the cell, and then what you have is a sporangium, and you have the forespore. All right, and there are two separate areas within the cell. The sporangium, this guy here, engulfs the forespore for further development, meaning it's going to give it another coating. And now you see here the blue coat and the yellow coat. All right, it continues to go through that process of adding layers to the endospore, and now you have that thickened cortex. Another few layers, all right, now it's looking like something's engulfing the endospore. It's completely engulfed. You have a mature endospore, and then the residual part of that mature cell is going to break away. All right, then you have the final germination process where, right here, you go back to a place where the nutrients are good, moisture levels are high, and then you go through germination. Germination is the process of moving that endospore back to a vegetative cell, and you can complete the process over and over again. So again, sporulation is not a reproductive process. It is a protective process. And so one vegetative cell can yield 2 vegetative cells if it's undergoing reproduction like normal. One vegetative cell can produce 1 endospore if it needs to create an endospore to save itself, and then 1 endospore can germinate to form 1 vegetative cell. So think about those numbers and the relationship between a vegetative cell and an endospore, and the distinction between reproduction and sporulation. And that is it for 3.4.